Good morning, students. Now let us continue the second lesson of Science 2 Life Processes in Living Organism, Part 1. Now, the next topic which we are going to cover is cell division, an essential to life process. Now, you can see on the screen some of the questions which are given. Let us go through the questions. First question: what happens to the cells of the injured tissue? After injury, the cells and the tissues are not able to perform the regular functions immediately. At the site of injury, the blood capillaries rupture and the area gets inflated. Many cells are damaged. The pain receptor nerves induce pain. Now, next question is there: whether new cells are formed during healing of wound. As the wound heals, there is scab developed. The cells surrounding the wound start dividing rapidly, and the cells lost in the injury are restored back. In this way, new cells are formed by the cell division to heal the wound. Now let us go to the third question. Do the plants get injured? When do we pluck the flowers? How are those wounds healed? It was thought that plants do not have nervous system, as they do not have sensations either. But recent discoveries have proved that plants also have sensations. The tissues that are lost get restored by cell division. Now let us go to the fourth question. How does the growth of any living organism occur? Does the number of cells in their body increases? If yes, how? Any living organism grows due to the increase in the number of cells in their body. The cells divide regularly and add new cells which are essential for growth. The cell division is thus necessary for the growth of the body and also for the regeneration and repair of tissues. Now, the fifth question: how are the new individuals of a species is formed from existing one or of same species? Answer: due to reproduction, the new individual is formed from the existing one. Reproduction can be asexual or sexual. In asexual reproduction, there is mitosis. This cell division helps in forming new individual. In sexual reproduction, gametes are formed by reduction division called meiosis. Due to chromosomes, genes and DNA, the new individual of a species becomes similar to the existing species. Now, let us see some of the important properties of living cell is cell division. What is the significance of cell division? There are three significance of cell division. First one, a new organism is created from pre-existing one. Second one, growth of a multicellular organism. Third one, restoration of injured and emaciated body. Now you all know there are two types of cell division. One is mitosis and second one is meiosis. What are the now let us go through the important features of mitosis and meiosis. Mitosis, it occurs in the somatic cells. Meiosis occurs in the germ cells. Okay. Next one, Mitosis, it takes place in two stages, karyokinesis and cytokinesis. And meiosis takes place in two parts, meiosis 1 and meiosis 2. Karyokinesis takes place in 4 stages and it is followed by cytokinesis. And meiosis 1 and meiosis 2 takes place in 4 stages, prophase 1, metaphase 1, anaphase 1, telophase 1, followed by prophase 2, metaphase 2, anaphase 2 and telophase 2. In mitosis, chromosome number does not change, whereas in meiosis, chromosomes number is reduced to half. In mitosis, mother cell gives rise to 2 daughter cells. In meiosis, mother cell gives rise to 4 daughter cells. Now the next one, the diploid cells are 2n and the haploid are n. All the normal body cells are always 2n, while only gametes formed by reduction divisions are n. In 2n condition, each type of chromosome is in pair, while in n condition, there is a single chromosome of each type. Now you can see the diagram of the chromosomes on the screen. Okay. Now these chromosomes, a pair of such type of chromosome is present in 2n condition, whereas single chromosomes of each type is present in n condition, and their structure like the one is shown in the figure, you can see. Okay. Now let us start with the mitosis. In the mitosis, we are going to see the stages of mitosis. There are 4 stages of mitosis. One is prophase, metaphase, anaphase and telophase. Now somatic cells and stem cells divide by mitosis. Mitosis is completed through 2 main steps. Those 2 steps are karyokinesis, that is a nuclear division, and cytokinesis, that is the cytoplasmic division. Karyokinesis is complete through 4 steps that I told you: prophase, metaphase, anaphase and telophase. Okay, now let us go through the 4 phases. First one is the prophase. In prophase, condensation of basically thin thread-like chromosomes starts. Why starts? Due to this, they become short and thick and they start to appear along with their pairs of sister chromatodes. Centroids duplicate and each centroid moves to the opposite poles of the cells. Nuclear membrane and nucleolus starts to disappear. Now second we are going to see is the metaphase. In metaphase, nuclear membrane completely disappears in metaphase. Chromosomes complete their condensation and become clearly visible along with their sister chromatids. All chromosomes are arranged parallel to equatorial plane of the cell. Special type of flexible proteins, they are called spindle fibers, are formed between the centromere of each chromosomes and both centroids. Next one is the anaphase. In anaphase, centromeres split and thereby sister chromatids of each chromosome separate and they are pulled apart in opposite direction with the help of spindle fibers. Separated sister chromatids are called as the daughter chromosomes. Chromosomes being pulled appear like bunch of bananas. In this way, each set of chromosomes reach at 2 opposite poles of the cells. Then the last one is the telophase. You can see on the screen all the phases, okay, of mitosis. In telophase, the chromosomes which have reached in at opposite poles of the cell now start to decondense, due to which they again become thread-like, thin and invisible. Nuclear membrane is formed around each set of chromosomes reached at poles. Thus, two daughter nuclei are formed in the cell. Nucleolus also appears in each daughter nucleus. Spindle fibers completely disappear. Now let us go through the cytokinesis. Now here we can see karyokinosis completes and cytokaryokinosis begins. The cytoplasm divides by cytokinesis and two new cells are formed, which are called as daughter cells. In this process, a notch is formed at the equatorial plane of the cell, which deepens gradually, and thereby two new cells are formed. However, in case of plant cells, instead of notch, a cell plate is formed exactly along the midline of the cell, and thus cytokinesis is completed. Okay, now let us go through the significance of mitosis. Mitosis is essential for the growth of the body. It is necessary for the restoration of emaciated body, for the wound healing, and for the formation of blood cells. Now let us go through the meiosis. Okay. Now meiosis is essential, is completed through two stages. These two stages are meiosis 1 and meiosis 2. Okay. In meiosis 1, recombination, crossing over occur between homologous chromosomes occur. This results in the genetic recombination. The four daughter cells formed after completion of meiosis are genetically recombined and not exactly alike, and also not exactly similar in their parent cell. Next, fourth one is the spores and gametes are formed by meiosis. Meiosis helps to restore and maintain the chromosome number. Now let us start with the meiosis 1. In meiosis 1, homologous chromosomes undergo crossing over, and hence there is genetic recombination. The homologous chromosomes are divided into two groups. From this, two haploid cells are formed. Prophase 1 of meiosis is lengthy phase, which is subdivided into 5 phases. They are leptotene, zygotene, pachytene, diplotene, and dikinesis. During pachytene, crossing over takes place. Now, meiosis part 2. Meiosis 2 is similar to the mitotic division. The two haploid cells that are produced in meiosis 2 now undergo further division, forming four haploid cells. Now let us go through the 5 stages of prophase 1 of meiosis. First only I told you the 5 stages of the prophase 1 of the meiosis are leptotene, zygotene, pachytene, diplotene and dikinesis. Okay. Now let us go one by one. Okay. In leptotene, the chromosomes start condensation and they become compact during leptotene. Next one is the zygotene. In zygotene, homologous chromosomes start pairing. This pairing is called synapsis. The structure called symptotomal complex develops to hold chromosomes in place during this pairing. Each chromosome's chromatid arm, divides, and form structures called bivalent or tetrad. Third one is the packeting. During packeting stage, crossing over of non-sister chromatids of homologous chromosomes takes place. Genetic recombination is produced due to such change. The homologous chromosomes still remained paired together at the sites of crossing over. Next one is the diplotin. During diplotin, the sypnotemal complex dissolves and the homologous chromosomes of the bivalent separates except at the point of crossing over. Thus, it looks like X-shaped structure called the chiasmata. Diagnosis. The last phase of prophase is for termination of chiasmata. The spindle fibers originate and the crossover homologous chromosomes are now separated. The nucleus disappears and the nuclear envelope breaks down. Meiosis 2 is just like a mitosis. Now during in this stage, the two haploid cells formed in the meiosis 1 undergoes division by separation of recombined sister chromatids and four haploid daughter cells are formed. Process of gamete production and spore formation occur by meiosis. In this type of cell division, four haploid daughter cells are formed from one diploid cells. During this cell division, crossing over occurs between the homologous chromosomes and thereby genetic recombination occurs. Due to this, all the four daughter cells are genetically different from plant cells and from the parent cell from each other